Good morning. Welcome to the Stillaguamish Tribe's Brenner Creek Hatchery. This hatchery has been in existence since 1923. It was called the Silverfin Trout Farm, and I found articles in the local Granite Falls Record from that time period, and the guy talked about how he had troubles with gardener snakes eating all of his trout. Fast forward, the Brenner family, Otto Brenner family, bought the facility in the 1940s, and they are the ones that expanded it. They also ran trout and provided trout for restaurants. They also provided eyed eggs and had a catch per inch trout fishery here in this large pond behind us. The tribe got the hatchery in 2010 as a result of the U.S.-Canada Treaty Annex in 2008. So we have a treaty between Canada and the U.S. that helps to figure out how we divide up the fisheries, because we have salmon from the United States mixing with salmon from Canada and Alaska, and those fish are caught by multiple nations, and so we have basically a treaty to figure out how we're going to catch those fish, how we're going to manage them, what kind of hatchery production. And as a result of that annex and how depressed the Stillaguamish Chinook runs are, provided the funding to purchase this facility. The reasons that we've got it are the run has been very depressed. We have the tribe's interest in protecting its treaty rights. With low salmon runs, they're not allowed to exercise their treaty rights on these culturally important and spiritually important fish, and so they want to see these runs rebuilt, as does everyone else that catches them and enjoys them. So in 2008, Canada and the U.S. governments provided the funding. We started building the hatchery in 2010, and first fish came into it in 2013. It's taken quite a bit of work and time. We're now in 2020, and we have a little more expansion work to do to have it completely finished. So anyway, what I wanted to tell you a little more about is the fish themselves. There are two populations of fish in the water, two populations of Chinook in the watershed: summer Chinook in the North Fork, fall Chinook in the South Fork. And originally, I started with the tribe 33 years ago. We were only worried about the summer Chinook in the North Fork. The reason for that is at the time, we thought all we had were bastardized hatchery fish in the South Fork, because the state of Washington Department of Fish and Wildlife had just been dumping excess hatchery fish from other watersheds, from the Skagit, from the Snohomish, from the Green River into the South Fork. And so we really didn't think we had a unique wild population in the South Fork. But with the ESA listing of Chinook, Puget Sound Chinook, NOAA said, well, we want you to look at the South Fork population anyway. So we went back and looked at historic genetic data, and we collected some additional genetic data. And lo and behold, we found out that we have a fairly clean, unique fall population of Chinook in the South Fork that look very different from the North Fork fish. And so I happened to be talking to George Pess at NOAA, who's a geomorphologist, and he says, wow, that makes sense geologically, because the North Fork used to have the Sauk and Suwallow River running to it at Darrington, and the South Fork Stilly used to run into the Pilchuck River in the Snohomish. And when you look at the genetics, the summer Chinook and the Stilly are most genetically related historically to the Sauk Suwallow fish, and the fall Chinook and the South Fork Stilly are most genetically related to the Pilchuck River fish and the Snohomish. So the genetics reflect the geology, and what changed that was the retreating of the glaciers. That changed the geological channels to what they are now. And so lo and behold, as much as we didn't really want to worry about the fall fish, NOAA said, well, you can't let them go extinct, and so we need to do something. And that is when the tribe petitioned the U.S.-Canada Treaty Annex scientists to give us funding to start this captive brood conservation hatchery. Again, this facility is different than our summer Chinook facility on the North Fork at Harvey Creek and at Whitehorse. That facility and those facilities, we go out, we just capture the returning adult Chinook in the North Fork, bring them back into the hatchery, rear them, and release them at the Whitehorse, WDFW Whitehorse hatchery, and then the juveniles return as adults. Because there were so few fish in the South Fork left, we had seen the run go from an average of 300 or 400 fish a year down to 80 and 100. We weren't able to capture the broodstock out in the river to start this facility up. So we had to actually go out and seine up the juveniles coming out of the gravel and rear those juveniles from fry size all the way to adult size, some 4, 5, and 6-year-old fish, to get the eggs to bootstrap the run. Because if we just caught the juveniles, brought them into the hatchery, grew them up, and released them, we wouldn't get as big an amplification as if we held all the eggs and reared the fish to full size so we could get the eggs out of the parent. So Brenner is a captive brood program. The Summer Chinook program is a traditional supplementation program. All right, so what I want to do is this facility is all spring fed. That is good for the fish in a number of reasons. It's relatively pathogen-free water. We don't have to worry about flooding. We don't have to worry about sedimentation. So we're going to go ahead and take a walk up, and I'll show you the springs and tell you a little about them, and then we'll come back and look at the hatchery. Okay, so we're up at one of the 2 spring sources for the hatchery. So when the silver fin trout farm started, they came up and they found these springs coming out of the hill, and they come out all over, and then we collect them with this screened box and then put it in a pipe down to the hatchery. When we were digging up, the contractor was digging up to put the new pipes in, we actually ran into some of the old cedar pipes that were used in the turn of the century to build the hatchery. We came in, they had just had a little earth embankment. I wanted to make sure we were capturing all the water. So I had the engineer put sheet pile in. Some of the sheet pile is down 60 feet to make sure that we captured all the water. Don Klopper, who used to be our water quality specialist, came up and tested the water, and he said, "Kip, this is some of the finest water I've tested in Western Washington. It is a sin that you're using to grow fish. We should be making bourbon with it." And so he was highly envious that the fish were getting water that he should be drinking. So we did all the tests on the water, and it comes out to find out we still had problems with our fish living in it. And it turns out that we have high CO2 levels in the water, which the trout farm people didn't deal with because their pipes were full of organic debris and biofilms, and then they weren't raising fish 5 and 6 years old. So we believe it's because we put the water into confined pipes, polyethylene pipes, and that were raising fish to 5 and 6 year old that we saw the problem. We've alleviated that by a blower system, which I'll show you, and then putting calcium carbonate stone in the springs along with oyster shell. We're spring dependent. When we don't get rain on the hill up above us, there are no mountains feeding this system. It's just the rain up on the flats. We see our water supply drop off. So droughts have impacted us heavily, especially 2015, 16. So we're in the process of putting a water recirculation system in to reuse the water to make sure we have enough to raise the fish. We have another spring over on the other side. We're not going to bother to look at it. It's identical to this one. So we're capturing about 450 to 500 gallons a minute when we're up at our normal flows. In 2015, we saw that drop in half, and that's kind of alarmed us. So, alrighty, so I think we'll head back down. The pipelines are buried. They go all the way down to the hatchery down this route. We are in the filter building, so the water comes from the springs, which I showed you, down through the pipes, comes into this building, and although the water comes from the springs, we still wanted to make sure that it was as pathogen-free as possible, because the springs can get frogs in them, birds poop in them. So we put in a system that we run the water through a 20 micron screen, and then it goes into the hatchery building where it goes through UV. So this is the whole process equipment that allows the water to turn on the filter, back flush it, turn it off, and then send the water into the UV in the hatchery. So that's what the purpose of this building is. So the water, I showed you the filter building where the water is filtered from the springs with a 20 micron filter. It comes through the wall and then it gets treated with these ultraviolet lights. And so we treat them with heavy UV ultraviolet to make sure that we can kill things like bacterial kidney disease and viruses that might be in the water. Again, it's unlikely because the springs are not connected to a river where fish are in them, but they can still have bugs and critters in them that we want to kill before we send the water to the fish. So this is the other half of that process. This is the ultraviolet treatment area. So we're here to show you about how we process the fish when they come in. As I said, we catch the juveniles out of the gravel because we can't get enough large fish like we do in the North Fork. Bring them in, adipose clip a little bit of, or caudal clip a little bit of fin off, and then we take that to the Department of Fish and Wildlife DNA lab, and they tell us if it's a true blue fall fish or a stray. If it's a true blue fall fish, we grow it up big enough until we can put a pit tag in it, and that tag is unique tag to that fish. So we know every fish's genetics in the hatchery and keep track of every one of them. We don't have to kill them with a radio tag. We can actually just pick the fish up and scan the nose of the fish or the stomach and see what the code number is and know which fish that is. And this becomes important when we start our breeding process to make sure that we're not crossing full siblings or half siblings. We want to keep, when we spawn the males and females, we want to make sure that we're not getting them too closely related to cause inbreeding depression. Once the fish are tagged, then they go into the tanks and we grow them out. They're tagged at about 120 millimeters. They go into the tanks and we rear them up until they're adult size. That could be anywhere from 2 to 6 years. After we reared them up, we have an ultrasound machine, just like what we use with humans to check the status of the growth of the fetus. We ultrasound the males and females to see how far along they are in their development and which fish will in fact spawn in the fall and which we hold till another year. Once the fish are spawned, then we put their eggs into these heath trays. Normally salmon lay their eggs in gravel, but we don't want to fill up these trays with lots of gravel, so we use a Vexar substitute. So we put the sperm and eggs together in this tray, lock the lid down like this, and then I'll show this to you again when we get in the building, and then water starts flowing through the tray and up around and goes to the next tray, and the water is bringing important oxygen to the eggs. Even though the eggs are not fish yet, they are alive and they can breathe across the membrane of the eggshell the same way that mom and dad fish use their gills to breathe. So they're alive, they have to have water. If we have water shut off for more than 30 minutes, we kill all of the eggs or alevins that are in the tray. Once the eggs are developed far enough along, then we can handle them. We can take them and shock them to show which eggs, are going to live and which are non-viable or dead. And then we can pick them and put them back in the tray. And then you can see the different developmental stages here. So these are the eggs that just been fertilized. The sperm had just gone into them, and they slowly develop. And you can see the eyes starting to develop on these fish. These two phases, the fish are so sensitive, if I bump the tray, I kill them all. Once they become eyed, then I can actually pick them up and bounce them. They're pretty bomb-proof as far as hurting them. And then they develop to what we call sac fry or alevins. You can see part of the yolk sac is still on the fish, and that's there to provide food. That's like the fish's umbilical cord, if you will. And then finally, they absorb and they become fry, and this is when they're ready to swim up out of the gravel or when we take them out of the tray. Once they're they're in here for about 2 to 3 months. They grow big enough. They become alevins or sac fry. They finally absorb the yolk sac, and then we move them out into the rearing troughs to grow them out till they're big enough to receive a coat of wire tag. The coat of wire tag is something that goes in their nose of the fish when they're about 2 inches long. So basically, this is a close-up of the coat of wire tags. They're about a 32nd of an inch long, thickness of a human hair, and they have these numbers etched on 3 sides of it. They make these tags on Shaw Island. They're the only company, Northwest Marine Technology. They have a worldwide patent on them, and how they managed to etch this number on that little teeny piece of wire is amazing. I believe they're using lasers to do it. So this wire again goes into the nose of the fish, just like you can put things in your ears or your nose, it doesn't hurt them. And then when we come back, we will wand the fish's nose with a unit similar to what we use with a pit tag, and that tells us that the tag is in the fish's nose, and we chop it out and read it, and it tells us which hatchery, which brood year the fish that we're spawning came from. All righty. The fish are tagged, then we move them out to the outside circular tanks. We rear them, and then they're released out into the river. And hopefully in 3 or 4 years, they come back. And when we get enough of our captive brood fish returning, then we hope to transition the program from a captive brood program to a regular supplementation program like we have in the North Fork. All right, well, let's go in. We also keep track of the fish, the adult fish that we spawn using jaw tags. So each fish that we know the DNA on, we also put a jaw tag on so that we look visually to make sure, again, that we're not crossing siblings or half siblings. We don't want inbreeding depression. So let's head into the hatchery. So basically, as I was telling you, when the baby fish that we get out of the gravel come into the hatchery, we take an adipose clip or a caudal fin clip to get the DNA, and they each go into their individual hotel. So we keep them in here until we DNA them, and then we keep them in here after we DNA them until they get big enough to move from these small hotels, we call them. They're actually little tanks. And normally they would be behind in this area right here. We have incubators in here right now, but this would be full of all these hotels, hundreds and hundreds of these that we keep the fish in. When they get big enough, and we've determined that they are true blue fall fish, then we move them from this hotel into these tanks and rear them up. So these are fish that were caught last winter. Were caught in February of this year, 2020. And now we've been rearing them in here until they get big enough to about 120 millimeters. And then we'll put that pit tag I showed you on the table into the fish's belly. Just like the coat of wire tag, it doesn't really hurt the fish. They seem to handle it just fine. And so we know the DNA, and we have a unique code for every fish that's in the hatchery. The fish are reared in here until they get a pit tag, and then they go into the tanks, the larger tanks. That have been pit tagged, they have that little tag in their bellies, and we're growing them out. A few of them, like the darker fish that you see down near the front, may actually provide, there he is, that dark one right there, they may actually provide sperm because that is a 2-year-old fish. And if it's a male, he may have sperm that we'll use in the breeding process. So we basically keep them together by brood year, and as they get bigger, we'll expand and split them into different tanks to make sure that they have enough room to grow. Now, the fish that started out as 1-year-olds are now 4, 5, and 6 years old. These are fish, as you can tell by their coloration, they're darkening, that are going to be spawned this fall in 2020. So these were babies that were caught in 2006, 2005, 2004. And so these fish are being spawned right now. We go through them every week. We'll take the eggs and sperm out of them. So these are all the females, and then we have a tank with males in them. That's the other tank that we showed you with the females, and these are the males that go with the fish in the other tank, and they'll also be spawned. Again, you can see they have the dark coloration. They don't have their ocean shiny silver and green backs like we see them out in the ocean. So we're in the spawning building. This is where the fish that were in those tanks that we showed you come to after we kill them. We bring them up here, and we put them on these bleeding racks. So one of the things when you're spawning salmon, you have to be careful about not getting blood or water mixed in with the eggs and sperm, because that deactivates them, and then you get poor fertilization and poor survival. So when we bring them in, we lay the females on one side, the males on the other. Each batch of eggs that's taken out of the female and each batch of sperm is put into a uniquely numbered bucket or container, so that we, again, can keep track of them genetically. We know this number is related to the pit tag number, and the pit tag number is related to the DNA number, so that we're making sure we don't cross sisters and brothers or even first cousins. So we're very careful about making sure how we keep track of who's who.
So I'm taking ovarian fluid samples, so that's the fluid that is around the eggs, and we take that back to our laboratory at Northwest Indian Fisheries Commission, and we test it for different fish viruses, and we use that information to help us understand what diseases are present in the watershed, and also it can help come to decisions about whether fish can move between facilities if tribes or WDFW want to transfer fish.
We'll hose them off to make sure they're nice and clean. They dry a little bit, and we dry them off with a towel, and we pick them up, and we'll spawn them into the bucket, and then also into the sperm container.
Beautiful.
Is it ovarian fluid okay though, right?
Yeah, no, it's just a lot.
Yeah.
Good.
What are you doing there, Garrett?
So I'm adding the fresh water, and adding the fresh water to the eggs and the milk actually activates the motility of the milk and opens the eggs so that fertilization can occur. I'm adding iodine to the eggs. These are fertilized and freshly rinsed, so they're nice and as clean as possible. And then from there we will add the iodine solution, which is exactly the same thing that surgeons use on cleaning human bodies prior to surgery. And what this does is just water hardens the egg and helps them give a nice, harder eggshell to better protect the developing fish inside.
And then those containers, once the sperm and eggs are together, get combined into the tray that I showed you before, and this tray gets put back into the rack, and that completes the cycle. But once the fish are spawned, then we put the eggs into those trays like I showed you. And you can see we actually have eggs. I'll show you very carefully. There you can see, this is the better tray. Not all the trays turn out. The pink eggs are alive, the white eggs are dead. And then there's a little bit of fungus growing on them. So these eggs were laid down on September 24th.